Hello everyone. So today in this video we are going to talk about tRNA or transfer RNA and about its function and its structure and all its basic information to advanced level information which would be enough for any national level examination including Neet or CSIR JRF or DBT JRF. So the information about tRNA is complete in this video. So let's talk about tRNA or transfer RNA. It is actually involved in the process of protein synthesis, right? It is involved in protein synthesis or what is known as translation. Now tRNA, as you can see, is the full form is transfer RNA. So what it transfers, as the name suggests, it transfers something. It transfers amino acids to be encoded into protein. It brings charged amino acid and it inserts them or transfer them into protein. So this is the function of transfer RNA or tRNA, right? So it is a small, well characterized RNA molecule with a key role in protein synthesis, right, or in translation. So let's see its structure. So as you can see in this structure, let me zoom in. Right, so as you can see in this structure, it has an acceptor stem with CCA is a conserved 3 unpaired sequences which is present in all tRNAs. Here is a T arm or what is known as T psi arm, right, because this contains thymine nucleotide. Psi is pseudouridine and C is cytosine, right? So this is T arm. It has also a D arm which contains dihydrouridine, right? And it has an anticodon loop and an anticodon arm. So this anticodon arm is what is connected to mRNA. It actually reads the mRNA, right? So mRNA will be somewhere here, and this anticodon loop is the structure that will actually read the mRNA and bring the specific tRNAs related to that amino acid, right? So let's move on to the next part. So it is also known as an adapter RNA. So what is an adapter RNA or what is an adapter? Let's first see. So for example, you take a charger or socket, you have a socket and you want to insert your charger within the socket to charge your phone or anything, but for some reason the shape between this socket and your charger is not compatible, so you can't actually insert, you are not able to insert this charger into the socket, right? You can't insert into it due to size difference or shape difference, right? So at that point of time, what you would actually do is you will buy an adapter, electrical appliance, an adapter. What will this adapter do? This would provide an interface between the socket and your charger, right? So it will provide a medium through which this socket and this charger can be connected. So this is the function of an adapter, right? So this is exactly what this adapter RNA, what is known as tRNA, does. The concept of an adapter provide interface between nucleic acid language and protein language. It was introduced by Crick in 1955, right? So let's see. So what it actually does is that our DNA contains some information, certain information in the form of genes or nucleic acid, right? Nucleic acid. So DNA has the information in the form of nucleic acid which needs to be converted to the protein which is in the form of amino acid language. So adapter RNA provides this interface between the DNA nucleic acid and the protein amino acid. The adapter thing provides or converts this nucleic acid information into the actual amino acid which needs to be encoded into a protein. So that is the function of an adapter RNA or tRNA. Right? It converts the DNA information of nucleic acid into amino acid which needs to be converted into protein. Right? So let's move to the next part. tRNAs also participate in non-translational processes such as during reverse transcription of retroviral RNA. It acts as a primer. So let's say, what is reverse transcription? So when DNA is used as a template for the synthesis of RNA, it is known as transcription. This is the general process of how RNA is transcribed from a DNA template. Now, if the reverse happens and RNA is used as a template for DNA synthesis, then that is known as reverse transcription. And sometimes tRNAs participate in reverse transcription. Right? So, that is something new to know about. Let's move on to the next part. Now, tRNAs is a single RNA chain of 73 to 93 nucleotides, mostly having 76 nucleotides present in the cytosol in a semi-autonomous organelle in all living cells. Right? So most of them have 76 nucleotides. Now Holy and his co-workers first determined the sequence of yeast TR in 1965. So in 1965, the actual sequence of tRNA of yeast was determined. Dictated by their primary sequences, tRNA folds into a clover leaf-like secondary structure with well-defined stems, loops that make up an acceptor D-arm, anticodon arm, and T. This is not TY arm, TYC arm, but this is T-Psi arm, right? This is T-Psi arm. So let's zoom into the structure and let's see. So this is the acceptor stem, which have ACC conserved sequences, are here amino acid come and join, right? At the acceptor stem, the amino acid come and join to ACC structure, the 3' end. This is the T-Psi, which has thymine for T, pseudouridine for psi, and cytosine for C. This is the T-arm. This is the D-arm, which has dihydrouridine, an unusual amino acid, and this is the anticodon arm and anticodon loop, which actually connects to mRNA and reads the mRNA. Right? So these are the loops and the stems that are actually present in the secondary clover leaf-like structure. So let's see why it's called clover leaf. This structure of tRNA that you see here is called clover leaf structure. That is because clover leaves look something like this, right? So this is a clover leaf actually looks like, and so this is how it was named as clover leaf structure in the secondary structure, right? This tertiary structure, which is actually functional, is much more complex than this, right? Let's move on. So here it says that regardless of the length of the tRNA, the numbering of conserved nucleotide remains constant, and the clover leaf-like secondary structures formed in virtually all tRNA. So there are about 50 types of tRNAs, 50 specific tRNAs for different amino acids in eukaryotes, and around 100 or something tRNAs in prokaryotes. So all those tRNAs have the same clover leaf-like secondary structure, right? Now let's move on. So acceptor arm always have 7 base pair and 4 unpaired nucleotide sequences, including an absolutely conserved CCA sequences. So this acceptor stem has 7 base pairs and 4 unpaired nucleotide sequences, in which CCA, here you can see the CCA at the 3' end. The CCA is conserved. because here is the part where the CC is where the amino acid come and join. Every tRNA is specific for one amino acid. Every tRNA is specific for one amino acid. For example, let us say that this tRNA is specific for glycine, right? Glycine amino acid. So this CCA will be connected to the amino acid GLY, glycine, right? Glycine will come and join here, and that glycine will be inserted into a protein or a peptide through this tRNA transfer RNA. So let's move on. So now 5' CCA 3' end is universally conserved for all tRNAs. Many genes that encode tRNAs do not encode the CCA end. Now CCA 3 at the 3' end is, as you see, universally conserved. It is required in all tRNAs. In prokaryotes, in eukaryotes, in all the 50 to 100 different kinds of tRNAs present in an organism, all that require this universally conserved CCA sequence. But certain genes that encodes this tRNA does not have the CCA end. So how the CCA end is added into it? A specialized RNA polymerase called CCA adding enzyme or tRNA nucleotidal transferase. This is an enzyme, tRNA nucleotidal transferase is an enzyme that adds CCA at the 3' end of every tRNA post-transcriptionally. After it is transcribed, at post-transcriptional process, this tRNA nucleotidal transferase adds the CCA sequence at the 3' end. Right? Now, as its name indicates, this enzyme adds the terminal CCA to the tRNAs that initially lack the sequence without RNA or DNA template. So this tRNA nucleotidal transferase, which is a polymerase enzyme, doesn't require any template for adding CCA sequence at the end of it. So let's move to the next part. Now here again, the structure of transfer RNA is shown. At first, this is a two-dimensional or secondary structure of clover leaf-like shape. This is what is called a clover leaf-like shape. This is clover leaf, right? So it has a D loop, which here you can see D. This D actually indicates dihydrouridine, which is an unusual nucleotide present in the D loop. So this is called D loop because it contains D or dihydrouridine. On the other hand, you can see the T-Psi loop, right? T-Psi loop is named T for thymine, Psi for pseudouridine, another unusual nucleotide, pseudouridine, and C for cytosine. So you can see Psi means here is the pseudouridine present, C means thymine present, right? And T means cytosine present, right? And this also shows a variable arm. So certain tRNAs also contains a variable arm, while some tRNAs does not contain this variable arm, right? So this is the secondary structure, the two-dimensional structure or the clover leaf model of tRNA. But there is the tertiary structure, how the molecule actually looks within the body or within the cytosol of the cell, right? The three-dimensional or the tertiary structure, the actually functional structure of the tRNA looks something like this, an L-shaped model. This is called an L-shaped model because it looks like an inverted L. So this is the anticodon loop. Here is somewhere the T-Psi loop. Here will be somewhere the D loop. And this is the 3' loop where CCA will be present. And the amino acid will be connected here. Right? So this is actually the three-dimensional structure, the tertiary structure of the tRNA. So let's move on. So the D arm or the 3 or 4 base pair stem, the D arm or 3 or 4 base pair stem and the 5 to 7 nucleotide loop that frequently contains the modified bases dihydrouridine. So D arm, like I said, contains dihydrouridine, which is a modified base. What it means by modified base is that uridine is a common base found in RNA, and this uridine is converted into dihydrouridine. So it is modified base that is added into the D arm. Okay, so that is an unusual base present. An anticodon arm with 5 base pair stem and loop that contains anticodon, a 3 nucleotide long sequence that is responsible for recognizing the codon by base pairing with the mRNA. Right, so let's go to the back to the picture again. Okay, so they were talking about the anticodon loop, right? So anticodon loop, as you can see here, CCG is present the 3 base pairs, CCG is the 3 base pairs present here, which actually reads the mRNA. It connects to the mRNA and reads the mRNA, and according to these anticodon, specific tRNA will come and join here and bring a specific kind of amino acid, right? So that's what we are talking about here. Now TYC, again I would like to say that wherever there is TYC, it's actually not TYC but T psi C. Okay, T psi C loop or T arm with 5 base pair stem and a loop that usually contains unusual base pseudouridine. So T psi C contains pseudouridine, another unusual base. Now variable arm usually has 4 to 5 nucleotides but can contain up to 24 nucleotides. Okay, so that's about variable arm of the tRNA. Now all tRNAs share the same secondary structure. tRNA is classified according to the length of their variable arm, which can be 4 to 5 nucleotides, class 1 tRNA, or 10 to 24 nucleotides, class 2 tRNA. So there are 2 classes of tRNAs, right? So there are 2 classes of tRNA. Class 1, which has around 4 to 5 bases in the variable arm, or class 2, which has around 10 to 24 bases in the variable arm. This is all in the variable arm. The variable arm decides which class of tRNAs it is, right? So right, that's about tRNA, class 1 and class 2. Class 1 with 4 to 5 nucleotides in the variable, and class 2 10 to 24 nucleotides in the variable loop. Now the next is a new class of class 2 tRNA discovered more recently in many organisms. Serves as adapter for selenocysteine in the 21st standard amino acid. So until now, many of us knew only 20 standard amino acid, but now there are 22 standards amino acid. The 21st amino acid, the new standard amino acid, selenocysteine, and a new class of class 2 tRNA has been discovered recently, which inserts selenocysteine, which is specific for selenocysteine, the 21st standard amino acid. So let's move on. Now Here you can see the secondary structure of a tRNA molecule folds into the L shape of three-dimensional conformation. So this is a three-dimensional conformation in the shape of an inverted L through complex tertiary interaction, including those between D loop and T psi loop. Depending on its sequences, the variable arm might form interaction with the D loop, right? So the variable arm forms an interaction with the D loop, right? So now there are some unusual post-transcriptionally added modification through enzymes which are different from normal bases in polynucleotide chain, right? So let's see them. For example, pseudouridine, like we talked about pseudouridine earlier in the T loop, is derived from uridine by an isomerization in which the site of attachment of uracil to ribose is switched from nitrogen at the ring position 1 to carbon at the ring position 5. So the position of the uracil with the base is nitrogenous base is changed from 1 to 5 to convert the uracil into pseudouridine. Right? So this is a post-transcriptional modification, right? Another unusual nucleotides found are dihydrouridine, derived from uridine by enzymatic reduction of double bond in the uracil between the carbons at the position 5 and 6. Some other unusual bases are also found in tRNA, including hypoxanthine, thymine, and methylguanine. Hypoxanthine is derived from adenosine and methylguanine from guanine. The tRNA is key to deciphering the code words in the mRNA. Each type of amino acid had its own type of tRNA which binds to it, carries it to the growing end of the polypeptide chain if the next code word on mRNA calls for it. So what's actually telling us is that there is an mRNA which encodes the mRNA which encodes the protein sequences, and every tRNA is specific. Every tRNA is specific for one amino acid, right? For example, one tRNA can be specific for glycine, another tRNA can be specific for valine, right? So that tRNA can only bring glycine to the ongrowing peptide, or if it's specific for valine, it could only bring a valine into the peptide, right? So tRNA through the anticodon loop reads this mRNA sequence, and if it encodes for valine, then a tRNA specific for valine will come and join here and transfer a valine sequence into the peptide. So that is the process of how it works, how translation works. Now, the prokaryotes contain around 30 to 45 different tRNAs, while eukaryotes have 50 or more different type of tRNA specific for different amino acids. Now, the standard amino acids in organisms in eukaryotes is only 22, but there are 50 different tRNAs, which means that certain tRNAs can bring the same amino acid, right? For example, if a tRNA, specific tRNA brings glycine, there could be other tRNAs as well that only brings glycine, right? So there could be more than one tRNA which brings the same amino acid. So two or more different tRNAs are specific for the same amino acid, termed as iso-accepting tRNAs, which means that there could be more than one tRNA which could bring the same amino acid, which means that this tRNA, more than one tRNA could be specific for the same amino acid, right? These are known as iso-accepting tRNAs. Now, for example, two tRNAs specific for glycine could be written as tRNA glycine 1 and tRNA glycine 2 because both tRNAs are different and yet they are specific for the same amino acid glycine. Thank you very much for watching this video. Please like and subscribe the video.